since I am the president, Wenching is aware of that, and so is Leah. If you guys can, attend the next year's ASIP meeting. Hey, how are you, AK? Aki? So
Where is the meeting next
it's in
year?
Portland,
Portland. It's in Portland, Oregon.
Oh, Oregon,
beautiful
very
place.
far.
Yeah. Well, I mean, for some it's near. Like for Eki, it's only a hour flight. For us, it's going to be definitely 3.5, 4 hours.
Yeah. Well, well, definitely, Baltimore is not a good place for us for the meetings.
Yeah. Well,
Yeah.
Baltimore, yeah, everybody. I mean, there were a couple of people from NIH, I think, and they just came for the meeting and then they went back. I think that's the problem, right? So if you are
Yeah,
local,
we cannot stay in hotel there. So
yeah,
we have to drive,
yeah. No, I think that's a problem.
well,
I saw Yan Xiao is also online. Paul Yan is a former postdoc in Mitch Lassa's lab. Now he's on the market and she's have some offer already.
Oh,
Yeah,
wow, that's
yeah,
great.
yeah.
That's
Do you single cell in the liver?
what's the name? What's the name?
Oh,
Do you want? Yeah.
oh, that is a institute in upstate New York.
Oh,
Uh,
wonderful.
oh.
What is it called?
It's called Masonic Medical Research Institute, but
Oh,
I'm still looking for still have some other interview going now.
okay. That's fantastic. That's fantastic. Well, yeah, keep in touch.
Oh, thank you.
Oh, you have your former trainee here. You want to
I have,
speak? Gang, Zheng Gang.
where is Gan? Is he here, George?
Yeah. He asked me about your Zoom.
Oh,
Yeah,
wow.
I'm here, but my video is not working. Good morning. Yeah,
So,
I'm
George,
here.
nothing has changed. So nothing has changed, huh?
Yeah,
Well,
maybe
good to
I'll
see
go
you,
to
bro.
Pittsburgh this summer to visit you. To visit you.
Please,
Yeah.
anytime, anytime. Yes.
Yeah.
Yeah,
So
I
now
was
is
hoping my student who's in Tsinghua University is finishing his fourth year of medical school. I don't know
Oh,
if they ever get the email or not. He was one of the best. Some of the data that I'll present today is actually from him, Shikai Hu.
Who? Yeah, I'm not sure
Shikai,
he's here.
yeah,
But
yeah.
Chiffon said I'm not sure. You know Chiffon many years ago, your trainee.
Yes, yes,
Yeah.
yes.
Uh, Dongying, I think it's time. You want to?
okay. Yeah. Okay, let me give an introduction now. So, yeah, I don't want to blow, so that you can have a conversation. Okay, so good morning, everyone in China, and good evening, people in North America. So it's my great pleasure to introduce record. I will deny for that. Okay, so it's my really great pleasure to introduce Dr. Pomanga to this, metabolic committee. Um, Paul earned his MD and also completed his internship in India, then follow with postdoctoral training at Temple University, that's in Philadelphia, same as I did postdoc at Philadelphia, but that's a lot of different years ago. He then moved to Pittsburgh till now. As Wenxin have a bigger, poster, have fantastic introduction for the Paul. I said some sentence here. He said that over the past 25 years, Paul has perspective investigator beta-catenin and winter pathways in liver development, regeneration, chronic liver diseases, and HCC. And I know recently work about this metabolic liver donation. His research has significantly advanced the field of basic liver biology and liver diseases. So, like 1.5 years ago, when I visited Pittsburgh, I was really very impressed by Paul showing me the data about this kind of spatial single cell transcriptomic data, and following with a very fantastic functional study show how like endothelial in specific zone affect hepatocyte in another zone manner. In my knowledge, I think this is the best functional zonation study in the liver. Paul also served in multiple leadership roles in the University of Pittsburgh, including funding director for the Liver Research Center. And now, as presented here, is the associate dean of research in Pittsburgh School of Medicine. The Liver Center is fantastic, include a lot of good PIs in Pittsburgh, but also in Cleveland, including our co-host Xiaoqing's postdoctoral mentor Laura Nagy there. And basically they cover all aspects in my mind to liver physiology and liver diseases. And when I visited Paul also gave me this cap. If you can write, see clearly, it's called Building Committee Links Like Livers. So different people have different interpretation. To me, like people are different place, maybe zonation have different functions, like different distinct cell type. So I think this word also linked to today's topic. I really eager to learn more about it, Paul. Thank you very much to being here, and the stage is yours.
Right. Well, thank you so much. I'm going to share my screen, and then we can get started. I'm just going to put this here, and I hope everybody can hear me and see me.
That's
right?
Good.
Okay, great. Well, first of all, really thank you, Dongyun, for giving this opportunity to be part of, presenting this talk data to esteemed colleagues here in the US and in China. Seems like it's been a very successful seminar series. So really grateful that I'm able to participate in it. Thank you for doing it. I think it also fosters nice collaboration between groups. So I hope that this will just be beginning of collaboration. I've visited China many times. It's a beautiful, beautiful place. And I hope to visit again, if I get an opportunity.
Recording in progress.
But it's been, it's truly, a great opportunity for me to talk about the kind of work that my lab has been doing. So the title of my talk is Role and Regulation of Hepatic Metabolic Zoning, and what are its implications in liver pathophysiology. So, so my group has been studying liver disease for quite some time. As Dongyun mentioned, I've been involved in liver studies all the way since my postdoc training in 1996. And then when I started my lab in 2001, I've just focused on liver development, regeneration, injury, and fibrosis and cancer. So why is it important? If you look at the global burden of liver disease, it's the 11th leading cause of death worldwide. And in the United States, it's actually 9th leading cause of death based on data from very recently from 2024. And of course, the liver can be infected by a spectrum of diseases, which then eventually result in its morbidity and mortality. And there is some geographical sort of association to the kind of pathologies. But overall, if you were to look all across the globe, I think alcohol remains one of the leading causes of cirrhosis globally, and its prevalence is even higher in high-income countries. And of course, right along alcohol is the non-alcoholic fatty liver disease, which is now labeled as metabolic dysfunction-associated steatotic liver disease, which also affects almost a quarter of global adult population, is mostly a disease of overnutrition. And of course, these alcohol and metabolic dysfunction don't have to happen mutually exclusively. So there is actually a new pathology that has been described in the recent nomenclature, and that's called MET-ALD, where both alcohol and metabolic dysfunction are playing an important role. And of course, we know that although HCV is potentially cured, other than that the drug is very expensive and not available to specifically inner cities and also rural parts of the world. But hepatitis B virus continues to be a major cause of liver mortality. And also, as the people are living longer and longer, aging itself is considered to be a cause of hepatic morbidity because aging liver becomes a sort of a common ground for inflammation, fibrosis, and lack of regeneration. And any of these pathologies can eventually lead to, cirrhosis and end-stage liver disease and liver cancer, which can really go hand in hand with advanced liver diseases. So, liver is extremely vital. We can't do without liver. And I think one has to be reminded that liver is really the site where all the portal blood, everything that we eat, we drink through the intestine, makes its way through the portal circulation into the liver. And the cells within the liver, the workhorse, are your hepatocytes. And these hepatocytes have to perform more than 500 metabolic processes, along with other functions like protein synthesis, all the coagulation factor synthesis, hormones, production of bile, which is critical for lipid absorption in the intestine. And also, while it's doing all these synthetic and metabolic functions, it also has to be responsible for detoxification of any xenobiotics, for example, any microbial products, any other toxins that make their way into the liver by way of the portal circulation. So liver is a really, really busy organ, and hepatocytes are always busy in trying to do these functional aspects. So if you were to compare liver to another organ, which is also derived from endoderm like intestine, liver doesn't really have any dedicated stem cells. It remains controversial, but if you were to look at intestine and you look at Ki67, which is the marker of S phase, you will see intestine base of crypt, as well as transiently amplifying progenitors are continuously positive for Ki67. And as these stem cells differentiate into these cell populations and then eventually into enterocytes, the proliferation is lower and lower, and the cells become more differentiated. But intestinal functions are mostly absorption. But hepatocytes, if you look at liver, the liver does not have this active stem cell compartment. And if you were to stain liver with Ki67, very few cells, 0.1 to 0.01% of hepatocytes are in S phase, suggesting that hepatocytes need to really function on the synthetic and metabolic functions as compared to intestine, where you renew your lining every 4 days. Liver really renews the whole organ once a year. So very, very low turnover. But if liver is injured, every hepatocyte has the capability of regenerating and proliferating. If the injury is to hepatocytes, if the injury is to the bile ducts, then the cholangiocyte, the second epithelial cell of the liver, is also readily able to proliferate and expand. So coming back to the functions that liver performs, which are very diverse and multiple, liver has come up with a very interesting sort of pattern to perform these various functions in form of division of labor. So hepatocytes are organized if you look under the microscope histologically, they are organized into zones 1, 2, and 3. Sometimes it's called periportal zone, mid zone, and pericentral zone. And this is the hepatocytes that are closest to the inlet to the liver, microscopic inlet, which is the portal triad. There's portal vein, there's hepatic artery, and there's bile duct. And as the blood is coming through the portal vein and through hepatic artery, it's moving through the sinusoids and is moving towards hepatic vein, and then it empties, goes into the systemic circulation. So hepatocytes that are organized around the portal tract have very different gene expression as compared to hepatocytes that are away from the portal triad and closer to the pericentral. And this is what we call as metabolic zonation. So this is a cartoon really depicting the same thing. Portal vein, hepatic artery, the blood comes into the sinusoid, mixes and leaves through the central vein. But the sinusoids are, of course, lined by sinusoidal endothelial cells, which are atypical endothelial cells. They have no basement membrane. They have big fenestrae, so there can be exchange of metabolic products and nutrients and xenobiotics between sinusoidal blood and hepatocytes. And then there are other cells like macrophages and stellate cells, which are also present in the liver lobule. But what is interesting is that these hepatocytes that are around the portal triad, they express very different genes and have very different functions as compared to hepatocytes that are towards the central vein. And for example, if you were to look at albumin secretion, most of the albumin is expressed in periportal, midzonal, and less so in pericentral area. Most of the xenobiotic metabolism genes are expressed in zone 3 and less so in zone 1. Gluconeogenesis-related genes are in zone 1, and glycolysis genes are in zone 3. Even within ammonia metabolism or protein end product ammonia that makes its way through the portal circulation, most of the ammonia is converted into urea in zone 1 by these hepatocytes. So all ureagenesis genes are located here. And then if there is still some ammonia left in this circulation, then the few cells around the central vein are capable of converting that ammonia to glutamine by condensing it with glutamate. And that's the function of glutamine synthetase. And that gene or that protein is only localized in hepatocytes around the central vein. So very interestingly, the whole metabolic, synthetic, xenobiotic, lipid, gluconeogenesis, carbohydrate metabolism is very nicely compartmentalized within the liver lobule. And this can also be seen by different ways. This is a technique that we use in our lab, which is called 100-gene panel. It's a single molecule FISH and for 100 genes together, and this is called molecular cartography. I'll go into the detail in a little while. But if you were to look at these 100 genes, you can see these are not homogeneously present across the liver lobule. Some genes are more here, some genes are more on this side. So just kind of making sure that we can all appreciate the heterogeneity. And I should mention here that each color is one gene and each dot is one RNA molecule. So you can see that all genes, there are multiple genes that are expressed at different levels across the hepatic lobule. And of course, this can be confirmed by protein staining as well. So this is immunohistochemistry. You can see CYP1A2 is zone 3 hepatocyte. That's your central vein. It's not there at all in the periportal area. Another protein called hepcidin or HAMP1 and 2. This is only present in mid-zone hepatocytes, but not in portal triad or around central veins. And lastly, this is just an example of another enzyme, another gene called CYP2F2. And CYP2F2 is only present in the periportal hepatocytes, and your pericentral regions are completely free of this particular gene or enzyme. So not only is the zonation play an important role in metabolic compartmentalization, it also allows repair process. And a simple example is, for example, acetaminophen overdose. If you have regular doses of acetaminophen, we know it can be conjugated and excreted. But if you have excessive acetaminophen, then CYP2E1 converts it into a toxic metabolite called NAPQI, which is responsible for DNA adducts, protein adducts, and leads to necrotic death of hepatocytes. Now, what is quite interesting is that you have your, if you look at where CYP2E is located, then that's the enzyme that converts acetaminophen into this toxic metabolite. It's there in zone 3 hepatocytes, and it's not there in zone 1 hepatocytes around the portal triad, and very little in mid-zonal areas. So if you take excess of acetaminophen, then most of the injury happens in pericentral region. So this is your central vein. You can see here, as marked by these arrowheads, this is where necrosis is happening, and all the cells around this area are still alive. And what happens then is that within 24 hours, there is a wave of cell proliferation, as can be seen by expression of cyclin D1, which is very important in G1 to S phase transition. And right away, Ki67, which again marks hepatocytes in S phase, begin around this necrotic area. And then within the next 24 hours, you can see the liver lobule is completely regenerated. So the functions of zonation go beyond synthetic and metabolic function. They also allow injury to be compartmentalized within a liver lobule so that the rest of the liver can regenerate and repair. And, you know, from the pathological perspective, if you were to look very closely at many liver diseases, you actually see that many liver pathologies are also zonated. I'll give you two examples. One, autoimmune hepatitis, and of course, our favorite metabolic dysfunction-associated steatotic liver disease, or MASLD. You can see here, autoimmune hepatitis is typically characterized by portal inflammation. This is your portal triad. Look at how many immune cells are present in this area, and interface hepatitis. All of this is happening in zone 1. Whereas if you were to look at MASLD, especially when MASLD starts, it starts usually in zone 3 around central vein. And as the disease gets worsened, the steatosis and steatohepatitis becomes more panzonal. So we have been interested for a very long time to understand what dictates this zonation process. And, what we have found is that a Wnt signaling pathway, there were 3 groups that really published the study almost around the same time in 2006. A French group, Sabine Colneau and Christina Perret, Matthias Hebrock and Sakine from UCSF, and our group, showing that the Wnt beta-catenin pathway is very important in playing a role in zonation process. So what is this Wnt signaling pathway? So I just sort of, for the sake of simplicity, just show here that there are 19 Wnt's. And all of these Wnt's need a protein called Wnt-less to be secreted from a cell. Once these Wnt's are secreted, they bind to their receptor frizzles, and there are 10 frizzles. Any Wnt can bind to any frizzled. And then there is a co-receptor, LRP5 or 6, that is also absolutely essential for Wnt signaling. So Wnt needs to bind to frizzled and co-receptor LRP5/6. This leads to inactivation of beta-catenin degradation complex. Beta-catenin cannot be phosphorylated. It's released from the complex, translocates into the nucleus, where it binds to this TCF family of transcription factors to turn on target genes. In the absence of Wnt's, beta-catenin degradation complex is active. Beta-catenin is phosphorylated at specific serine and threonine residues, which makes it recognizable by beta-TRCP, which then allows for ubiquitin proteasome-mediated degradation of beta-catenin. So again, any of these 19 Wnt can bind to any of the frizzles and LRP5 or 6, resulting in beta-catenin activation. So how did we find that beta-catenin is actually playing an important role? Obviously, the first thing that was done back in 2006 was to knock out beta-catenin. So we made a flux beta-catenin animal and deleted using albumin Cree. And as you could see, if you look at some of the traditional zone 3 genes, CYP2E1, CYP1A2, and glutamine synthetase, those are completely lost in the knockout on the right. This can be confirmed by molecular cartography. You can see the pattern of gene expression using 100 genes is very different between the control and the knockout. So when you don't have beta-catenin in hepatocytes, you do not have zone 3 targets. So the question that we asked next was, is this beta-catenin activity under the control of Wnt signaling pathway? Because many, many different factors can control beta-catenin activation, including receptor tyrosine kinases like EGFR and MET and many others. TGF-alpha can control, FGFs can control. So what we decided to do, because it's difficult to knock out all 19 Wnt, it's difficult to knock out all of these frizzles, but because we only had 2 LRPs, so we conditionally deleted LRP5, 6 in hepatocytes and then checked zonation. And what we find is exactly the same thing that happened to beta-catenin knockouts, that all these hepatocyte LRP5, 6 knockouts showed lack of zone 3 targets, as shown for GS, CYP1A2, and CYP2E1. And this was also confirmed by Western blots. So that told us that that beta-catenin activity seems
Recording
to be,
stopped.
is supposed to be under the control of the Wnt signaling pathway. So then the big question for us was, where are the Wnt's coming from? Which cell type in the liver are the Wnt's coming from? Cholangiocytes? Are the hepatocytes producing Wnt's that are working in an autocrine fashion to regulate gene expression here? Are macrophages producing this? Are these endothelial cells that are lining these vessels, or are the stellate cells or fibroblasts of the liver? So what we decided to do was that we decided to knock out this protein called Wnt-less. Now, remember that if you don't have Wnt-less, then these cells that do not have Wnt-less are unable to secrete any Wnt's from that cell. So then what we decided, we had Wnt-less flox mice, and we bred them to various different Cree lines. We used albumin Cree to knock out Wnt-less from hepatocyte cholangiocytes, Live1 from endothelial cell, LRAD for stellate cells, and Lysem for macrophages. And the idea was that once you delete Wnt-less from these cells, then none of these cells would be able to produce Wnt's, and we will be able to compare in which phenotype, which genotype we have lack of zone 3, just like beta-catenin knockouts and LRP5, 6. And I'll just give you a positive example of, it took a lot of work, but what we found was only when you knock out Wnt-less from endothelial cells of the liver do we get the same exact phenotype as our other phenotype. And you see here, glutamine synthetase is gone, CYP1A2 and CYP2E1 is gone, and this is Western blots. So again, the overall message was that the Wnt's are coming from endothelial cells, which are then in a paracrine manner activating hepatocyte LRP5, 6, hepatocyte beta-catenin, and regulating targets. So, of course, now we knew the cells, and the next question for us was, which are those, out of those 19 Wnt's, which are the real Wnt's? So, by the time we were doing all of this analysis, Roel Noos's group, Bruce Wong, published in situ hybridization results showing Wnt2 was present in these endothelial cells lining the central vein and also sinusoidal endothelial cells. Wnt9b was also shown to be present in these cells. So we confirmed, so Shikai, when he was a graduate student in my lab, he used a single cell atlas that was published in Cell in 2022 to look at the expression of various Wints, and not just Wnt2 and 9b, but what he found was that if you were to look at Wnt9b or Wnt2 in humans, primates, pig, or mouse, the highest amount of Wints, 9 and 2, were expressed in endothelial cells of the liver as compared to any other cell type. So that gave us confidence that perhaps Wnt2 and Wnt9b truly are the real Wints. So we also looked directly in our own single molecule FISH study, and where we were able to show in a liver lobule that Wnt9b is present in endothelial cells that are lining the central vein and in sinusoidal endothelial cells, mostly in zone 3 of the liver. How can we say that these cells are endothelial cells? Because we immuno in situ hybridization, we also did for PCAM1. And you can see here that this white cells are the PCAM positive cells, and there is consistent overlap between both Wnt9 as well as in Wnt2. Wnt2 is in red. Wnt2 was present mostly in sinusoidal endothelial cells in a gradient, which were present in zone 3, some in zone 2, but closer to portal triad, there was hardly any Wnt2. So there was very little overlap with macrophages and with stellate cells. So with that in mind, we decided to generate Wnt2 and Wnt9b double-floxed mice, and then bred it to live one Cree with a reporter, EYFP, stop rosa. And you can see when we breed these animals, we see EYFP. This is immunohistochemistry for EYFP, showing very nicely that we have been able to delete both Wnt9b and Wnt2 in endothelial cells of the liver, around central vein, in the sinusoids, also around the portal vein. And the question was, what is the zonation looking like? And for that, we decided to do single-cell spatial transcriptomics using molecular cartography. So we collected Wnt2, Wnt9b knockout livers and their genotype controls, and processed the livers in a specific manner, sent it to a company called Resolve Biosciences, gave them a list of 100 genes that we wanted them to make probes for, and then with each probe, they hybridized, they developed the color, they imaged, then they decolorized, then did this for 100 genes. And eventually, they sent us this type of a slide with all of this information. So again, you can, there is no way to do protein staining yet, but I think they are coming up with new technology in the next 2 months where you can actually add 30 to 50 antibodies on this section as well. But when we did this back in 2021, this was not the case. So we had to use AI. We used a program called QuPath to do cell segmentation. You give the rough size of a cell, and there is obviously a nuclear counter stain in blue on this section. So this AI program is able to then draw hepatocytes around the nuclei, and then once that information is done, we are able to calculate expression of 100 genes per cell. Now, remember, if this is one cell right here, all 100 genes are expressed. So we can identify the expression of these 100 genes in each cell, and then we can do unbiased or biased clustering to see how many clusters of hepatocytes are there in a liver lobule based on their gene expression. But what is the beauty is that because we know each and every X and Y location of this hepatocyte, we are able to then cluster map back to the tissue section. We call it C-maps, which gives us spatial localization of each cell within the cluster. So I'll show you what we did. So we used 16 genes to do a biased clustering for our Wnt2, Wnt9b knockouts and controls. So in a control liver, if you use these 16 genes, you can see that you get 6 different clusters in a liver, in a liver, C1 through C6. Now, because we know the spatial localization of each of this cell or, within the cluster, we can reverse map it to tissue section. So this is the tissue section from which this information was derived. And now we are able to make a C-map on this particular section. And you can see here the C1 is mapping to hepatocytes around the central vein. C2 is also in zone 3, but somewhat distant and distinct from the C1 cluster. Then your C3 and C4 clusters are mostly mid-zonal, and then C5 and C6 are mostly periportal clusters. So this is how your control liver lobule in a control mouse looks like. But what happens when you knock out Wnt2 and Wnt9b from these animals? What we find is that there is complete loss of C1 and C2 cluster, as you can see here, and there is enrichment of C4, C5, and C6 cluster. We can again generate a C-map, and when you project these clusters back onto the tissue section, what you see is that your hepatocytes that are surrounding the central vein now are mostly C4, C5, and C6. Your C6 cluster still is mostly periportal, but your C4 and C5 clusters are now present in your zone 3. What has happened is that the zonation of the liver is completely altered, and you are getting periportalization as well as mid-zonalization of the entire liver lobule, which was quite interesting to us. And this wasn't just true for Wnt2, Wnt9b double knockouts. We then examined every other knockout that we had generated over the last 15 years. And I won't show the C-maps and U-maps from those animals, but I'll just show some representative single molecule fish. So you can see here on the left, this is a control animal. CYP2E1 is marking your cells in the zone 3. And then CYP2F2 is mostly in zone 1, and partly, and CYP2E1 and CYP2F, they overlap in zone 2. And these are high abundance genes. And if you look at low abundance genes like glutamine synthetase, LEC2, and ragucalcin, you will see this is how your zone 3 looks like in a normal liver lobule. And then arginase 1 and ASS are periportal genes, and they are very nicely located in the periportal region. When you knock out beta-catenin from the liver, you can see that CYP2E1 is completely gone, and most of your liver lobule is now CYP2F2 positive. And the same thing happens here. All these blues, glutamine synthetase, LEC2, ragucalcin is gone, and your entire liver is now positive for arginase 1 and ASS1. So your periportal and mid zones have extended all the way towards the pericentral. Same thing happens with LRP5/6 double knockouts and with endothelial cell Wnt-less knockouts. So without the Wnt signaling pathway being present, I think your entire liver lobule becomes mid-zonal and periportal. So to us, this also means that zonation is dynamic and is a function of both transcriptional activation and repression of target genes at the same time. So we also started to ask the question, what is the role of Wnts in any, if any, in the hepatic stellate cells of the liver? Now, we all know that stellate cells are the fibroblasts within the liver lobule, and usually most of its function is known in collagen deposition. So if you have chronic injury to the liver, the stellate cells get activated, and then they will, undergo they will produce a lot of collagen and matrix for wound healing process. And a lot of work has been done by many of you on the call today in this area. So the question that we wanted to ask was, what is happening if you are removing Wnt from stellate cells? So we generated a Wnt-less floxed mice and bred it to LRAT3. LRAT was generated by Robert Schwabe and made a stellate cell Wnt-less knockout. And what we found was that the animals had smaller body weight. So this was very interesting to us that they have smaller body weight. We did echo MRI to see what the ratio of their fat mass versus lean mass was. And what was very exciting to us was that these animals were not only less in body weight, they had less body fat overall and had more lean mass. And when these animals were put into metabolic cages, they were less active, but they were showing more energy expenditure, although their food intake was normal and comparable to the controls. So less body fat, less activity, more energy expenditure, but they were eating the same. So quite a remarkable phenotype. So what we did was we did simple single nuke sequencing on these livers to kind of get an idea of what is different in these animals. And we just by unbiased clustering, we found many clusters. And when we looked globally at the differences of what were these knockout enriched in, these knockout hepatocytes enriched in, what we found was that these knockouts were expressing genes that were enriched for fatty acid metabolism, bile acid metabolism, xenobiotic metabolism. These were the top hits. And when you look at these different clusters, what we find is that there is a significant decrease in the knockouts of this cluster 1. And this cluster 1 is enriched in genes which are usually expressed in mid-zone of the liver. And they are increased. Like if you look at this cluster, which is cluster 8, this increases fourfold. And if you look at this cluster 8, this is enriched in very typical pericentral genes like glutamine synthetase, CYP7A1, OAT, and others, suggesting to us that there may be an enhancement of zone 3 at the expense of zone 2 in these knockouts. So because we already had our 100-gene panel for molecular cartography, just like what I showed you with Wnt9b, and Wnt2 double knockouts, we then subjected our knockout livers to this type of molecular cartography. And you can see here that, we found there was significant differences in the number of cells within each cluster when you compare wild type to the knockout. And when we were able to make C maps, these were the clusters that were present in zone 3. And you can see here, and these were the clusters that were present in zone 1. So you can see in a control animal, you have very nice zone 1 and zone 3. I'm just thinking if this is actually, did I flip it? I did actually flip it. Sorry. This is zone 3. So this color right here should be zone 3. This is zone 1. So I do apologize for this. This is zone 1. This is zone 3. And you can see here that in the knockout, there is expansion of these warm colors at the expense of these cold colors, which are these colors in blue. So very interesting and completely opposite to what we had with our beta-catenin knockouts, Wnt2 9b knockouts. In stellate cell Wnt-less knockout, there is enrichment, it almost seems like, of zone 3 at the expense of other zones. And again, we can do clustering on molecular cartography data. And what you can see here is if you look at cluster 0, the cluster 0 is almost more than doubled in the knockouts. And when you look at the genes that are enriched in this cluster, these are genes like CYP2E1, which are typically present in zone 3. Another cluster is this cluster 5. Cluster 5 is enriched about, from 7.2 to 18.3%. And this cluster 5 is also enriched in many genes that are localized in zone 3. For example, CYP1A, CYP2E1, OAT, LEC2, CYP8B1, and others. And a lot of them seems to be Wnt beta-catenin targets. And if you look at clusters that are decreased, like cluster 1, which goes from 22.6% in the control to 9.2, this cluster is enriched in periportal genes like arginase 1, CYP2F2, and ELOF2. So this was quite interesting, and we wanted to validate that by immunohistochemistry. And I hope this would convince you. This is your control animal, and this is your knockout animal. And if you look at CYP2E1 and CYP1A2, there is a wide of number of cells that are positive for these two markers. If you look at zone 1 marker like CYP2F2, it's considerably shrunken in the knockouts. And if you look at some of the mid-zone markers like hepcidin, they are completely almost obliterated. There's just elimination of this particular zone. So since we also had some non-parenchymal cells in the cluster, we were able to annotate them in our molecular cartography data as well. And we had markers for cholangiocytes, we had marker for endothelial cells, and marker for stellate cells. So we were able to do that and then project them back using the CMAPs. And what you can see is when you compare the wild type and knockout, again, you can zone 3 is wider as compared to zone 1, but then you can start to look at the different types of cells. Now, what is the beauty of this system is that we know spatial information of these non-parenchymal cells relative to each other and relative to hepatocytes. So we can use algorithms like CellChat to identify cell-cell communication. And that's what we did next. Since in a 100-gene panel, we had expression of we had 19 Wints, we had 10 Frizzle receptors, and we also had LRP5 and 6. We were able to address the probability of cell-cell communication using this program called CellChat2, which identifies a potential communication between the cell based on which cell is expressing a ligand and which cell is expressing the receptor, and taking into account distance between those two cells. So again, if you were to look in wild type, the y-axis has all the sender cells and x-axis has the receiver cells. And if you just focus on stellate cells, because in this model, we are knocking out the ability of stellate cells to secrete Wints. So you can see here zone 1 stellate cells seem to be communicating with zone 1 endothelial cells. So the higher the color, light 0 or white is 0, but as you go to dark yellow, red, brown, it means the probability of communication is higher. And you can see here that zone 1 stellate cells are communicating with zone 1 endothelial cells in zone 1, and in zone 3 also. If you look here, then zone 3 stellate cells are also communicating with zone 3 endothelial cells as well as with stellate cells. But what happens in the knockouts, you see that this signal is almost completely gone. There is no zone 3 communication with any of these endothelial cells. And also in zone 1, there is no communication of these stellate cells, which means also validates that our knockouts are not doing any communication at all. And then we were able to go spatially deeper into this and identify what are the signals that are missing. And this row is wild type and this row is knockout. And if you look very carefully, you can see what are the communications that were downregulated in the knockouts, especially in stellate cells. it were Wnt4. So Wnt4, and then there is a little signal from Wnt10A. But these Wnt4 communication to endothelial cell is completely eliminated in our knockouts. And to confirm this, we were able to present this data in a ridge plot format. And you can see again that this is your y-axis, this x-axis. This tells you the expression level of your gene. And you can see many cells have no expression of Wnt4. But if you look at zone 1 stellate cells, they have expression of Wnt4 gene, which is relatively high. Other cells express it are cholangiocytes and very few immune cells. But very interestingly, and then in zone 3 stellate cells also have a little bit of Wnt4, but not much. But in the knockout, this particular peak is almost eliminated, suggesting again that the knockouts that cannot secrete Wnt4 probably are able to downregulate this Wnt4 from the cells. Now, what were the communication that was up in the knockout? When you look at that, we only found Wnt2 to be significantly upregulated in the endothelial cells, which was also very interesting, that when you are knocking out your Wnt-less from stellate cells, then it almost is some other Wnt is coming up in your endothelial cells. And again, when you spatially look at it, you find it's the zone 1 endothelial cells, which do not usually express Wnt2. Now, remember, Wnt2 is always expressed in endothelial cells in zone 3. And this continues to happen in wild types and knockouts, but only in Wnt-less stellate cell knockouts do you see resurgence of Wnt2 in your knockout zone 1 endothelial cells. So this almost suggests that Wnt4 from stellate cells is inhibiting the expression of Wnt2 in this region. To confirm that this is truly happening, we looked at the single cell sequencing from stellate cell knockouts, focusing only on the endothelial cell compartment. These are all endothelial cell clusters. And what we find is that in knockouts, there is enrichment of this cluster 8, which is enriched in many markers like KIT, FABP4, and Wnt2 in pericentral endothelial cells. So these are markers of pericentral endothelial cells, and these are the ones that are increased in our stellate cell Wnt-less knockouts. So to confirm this at a protein level, we picked one of these proteins, FABP4. The antibody works very nicely. You can see in a control animal, FABP4 is expressed mostly in zone 3 endothelial cells in a control liver, but in the knockout, you almost see panzonal FABP4. Every single sinusoidal endothelial cell in the liver lobule is showing this expression. To us, this suggests that the whole liver lobule is now beginning to express zone 3 endothelial cell markers, which may be the reason why your pericentral target genes are now being expressed in mid-zone and even in periportal region at the expense of the normal genes that are usually expressed in that area. So this is just a cartoon sort of showing that we think in a control, Wnt4 is inhibiting Wnt2 expression in periportal sinusoidal endothelial cells, but in its absence, Wnt2 is expressed all the way up to here, along with other markers like FABP4 and c-KIT and others. And my last 2 or 3 slides, I wanted to just focus on what are the functional consequences of this expanded zone in metabolic zone on the different functions. So remember, one of the first gene enrichment, the gene ontology pathways that was upregulated was bile acid metabolism / synthesis. In fact, when we look at overall bile acid pool, the knockouts had significantly greater bile acids as compared to the wild types. These are total hepatic bile acids. I already said that, and the second big hit was fatty acid oxidation was increased in the knockouts. And again, decreased body weight was relevant to see. And remember that these animals by echo CT scan had echo MRI rather had less body fat as compared to lean mass. And then we took these liver slices from controls and knockouts and did Ouroboros, which is like a seahorse. And it was interesting to see that there was no difference in oxygen flux representing ATP production when you were using pyruvate as a substrate. But when you were using lipid like palmitic acid as a substrate, the knockout livers preferred and produced more oxygen flux here. And to us, this indicates that the knockouts are more proficient at using lipids as a substrate for energy production as compared to the carbohydrates. So based on all of this data, we decided to take these knockouts and challenge these animals with high-fat diet or with Western diet. And although the number is small, I just wanted to share this. We had, 3 controls and 3 or 4 knockouts. And what we saw is that all the controls gained body weight as compared to all the knockouts, which not only did not gain weight, actually they lost weight. And unfortunately, 2 out of these 4 animals died while within 2 to 3 months of the Western diet. But the 2 animals that still survived, when you look at their livers, you can clearly see that there was no fat accumulation grossly. And when you do histological examination, the controls had very nice pericentral and mid-zonal steatosis, whereas the knockouts, the ones that were alive, had absolutely no lipid accumulation in their livers. So, and then the final question was, okay, so this was metabolism. What happens to their regeneration process? Now, remember that most proliferative zone in a liver lobule is zone 2 and zone 1. Hao Zhu's work has shown that, Yan's work has shown that, and other groups from Shanghai have shown that, that zone 2 and zone 1 have a stronger proliferative compartment of the liver. Now, remember in our Wnt-less stellate cell knockouts, this particular zone is completely shrunk, and the CYP2E1 is actually expanded. So when we gave acetaminophen 300 mg per kilogram, what we found was that there was significantly more CYP2E1, there was significantly increased necrosis, and at the same time, there was very little cyclin D1 to actually no cyclin D1, and there was very little proliferation in these animals. So there was significantly more injury in these knockouts as compared to the controls. And finally, we also wanted to use another model, and this was partial hepatectomy model. We used, when we took knockouts and 2-month-old knockouts, and did partial hepatectomy, every animal died within 24 hours of surgery. But if you waited for up to 3 months, when these animals were 3 months old, and then did partial hepatectomy, we found significant decrease in the knockouts of cyclin D1 at 40 hours. But at 72 hours, cyclin D1 came back comparable to the knockout. And Ki67, again, at 40 hours, no Ki67, but at 72 hours, it kind of bounces back. So just to sort of summarize everything, Wnt2 and Wnt9b from endothelial cells are responsible for activating beta-catenin and are really playing an important role in division of labor or zonation by activating zone 3 gene expression. And loss of endothelial cell Wnt beta-catenin axis leads to expansion of midzone and periportal gene expression in pericentral hepatocytes. And Winds from hepatic stellate cells may be playing an important role in patterning zone 1 sinusoidal endothelial cells by restricting zone 3 endothelial cell expression, and in turn controls metabolic zonation of hepatocytes. Hence, I think it's important that spatiotemporal balance of factors like Winds, not excess and not completely eliminated, is really desirable for optimum biological function, including metabolism, repair, and others. And this balance, I think, is very important in homeostasis. So I'll just like to really thank the folks that have done a lot of the work that I presented here today. I think all the Wnt2, Wnt9b was done by Shikai Hu, who's back at Tsinghua University. Now he was with me for 3 years. And most of the stellate cell work that I showed you was done by Anya and by Tyler. Both of them are MD. Tyler is actually an MD PhD student. I did not get a chance to, and these are the pictures. So Tyler is here, Anya is here. And I did not get a chance to talk about liver cancer work, which is a whole another talk, but liver cancer zonation work is done by Brandon, also by Tyler, Evan, and Junian, who is actually the master of all the hydrodynamic tailwind injections that we do for generating our cancer models. So I'll really stop here, and we'll be very happy to have a good, robust discussion on the topic. Thank you.
Thank you. Thank you so much, Dr. Moga. It's really comprehensive and beautiful work. What I feel is like our zonation is like, it's really difficult to study.
It is. It is. And especially, and it's relatively easier in mice, but if you were to do this in patient samples, you can imagine how hard it is, because we have to learn about the circadian rhythms, which a lot of you are interested in. So, in mice, it's clear, but at least that's a good starting point to understand at least the basic biology and the circuits. And then we can start to kind of apply it to patient samples as well.
Yeah, exactly. If we relate it to the disease and human samples, it's more comprehensive and complicated. Yeah. So, well, now it's a Q&A session. Let's start with the panelist first. Dr. Dean.
Oh, I start first? Okay.
Yeah.
Oh, you know what? The peak time you have 350 people. I don't know, 250 people, I think. Okay. It's more than the ASIP we had the meeting, this week.
Oh, that's great. No, this is great. I really appreciate, again, the opportunity.
I hope most people, if you're interested, next year ASIP meeting is called American Society of Investigative Pathology at Portland, Oregon. You guys can directly in person interact with Dr. Manga. I have 3 questions. Paul, by the way, great talk as always.
Thank you.
Some of your content I have heard 3 times a week. I still have questions.
Yes.
I have 3 questions. The first question is, your second part, you mentioned about the stellate cell, endothelial cell. So in addition to hepatocyte zonation, apparently stellate cell or non-parenchymal cell, let's say that, also has a zonation. And as your data showed, different place of a stellate cell or had a single cell different Wnt expression. Can you comment on, so how does non-parenchymal cell zonation is regulated? Why they have different Wnt expression in different place of stellate cell and the single cell?
That's a wonderful question, Wenxing, and I think first of all, I don't think anyone has shown that Wnt4 is present in zone 1 stellate cells only. So this is the first time, and the paper is actually out now and is being reviewed. So we are hoping to hear from them. We don't know the mechanism. So I personally think that one cell type will regulate the zonation of another cell type. For example, I believe that the endothelial cell Wnt dictate hepatocyte gene expression in that zone. And I think hepatocytes are talking back to these endothelial cells or to macrophages in that area, right? So now that we have at least the Wnt beta-catenin models, we are looking at single cell sequencing to look at all different types of cells to see if just knocking out beta-catenin from hepatocyte does the zonation of endothelial cell, macrophages, stellate cell change. And that's very much work in the process. But I think that there will be some hidden circuits that we have not been able to appreciate, which are coming back and forth from these different cell types.
I see. Thank you, Paul. My second question is, I'm wondering, maybe you can comment whether a new trend change, for example, fasting feeding in the liver, will affect the metabolic, apparently it will affect the lipid glucose metabolism. Does fasting refeeding will affect the Wnt signaling, then affect the liver hepatocyte zonation?
Yes, so it's a great question. And we published actually Jay Bihari, who was a fellow long time ago, and now he has 2 R01s of his own. He's a associate professor of medicine, gastroenterologist. He published a couple of papers where he did fasting refeeding and looked at Wnt signaling pathway. And there were significant differences in how Wnt change during fasting refeeding. Now, I should mention that we did not have the sophisticated technologies of zonation-specific changes other than just looking by PCR and other things. So I think now we are going back to those types of data and looking at it through the lens of molecular cartography or Visium, and we'll be able to now see if the fasting refeeding is actually changing the Wnt signaling in specific zones of the liver. And my hypothesis is definitely it's going to be, obviously you have to control for circadian rhythms and 12-hour cycles, but if you can go beyond that, I think there would be definitely differences. We have not gotten to that point yet, but it's very much in the next 5 years, we're going to be focusing on that.
Okay, great. My final question is, so actually I have to came back early for my SIP meeting because Robert Schwabe visitors. So he gave a talk. He mentioned some of your data. So in some of his data, he showed when he knocked out the stellate cell sponding.
Yes.
So that's with the LGR pathway. My question is, what's the relationship of Wnt fizzle signaling pathway and this sponding pathway? Does these two pathways have any compensatory effect?
So no compensatory effect. So think of it as the Wnt are the big brothers and the R-spondins are the small sisters. So I think what R-spondins are doing are really fine-tuning the pathway. So when you knock out R-spondins, you will never get a black and white phenotype. Whereas you knock out Wnt beta-catenin, the phenotype's going to be black and white.
I see.
So if Wnt signaling is there, knocking out R-spondins will only have a white effect. So when he knocks out R-spondin, so if you remember, Jan Chors from Novartis had knocked out R-spondin. Glutamine synthetase doesn't, the 2E1 doesn't disappear. It decreases, right?
Okay.
And now they knock out, Robert Schwabe knocks out R-spondin from stellate cells. And when they knock it out from stellate cells, again, you don't see complete changes in zonation. You see decrease in there because it is fine-tuning, the Wnt signaling pathway. So for optimum signaling, you require R-spondins and Wnt's both to kind of act in almost, I wouldn't call it synergy, but at least it's just optimizing the Wnt's to be.
I see.
That's it.
I see. Oh, great. Thanks so much, Paul. Great talk. Thank you.
Thank you, Wen.
Thank you, Wenxin. Charlie?
Yes, thank you, Xiaoqing. Paul, great talk. Really enjoy.
Thank you.
Yeah, I have a couple of questions. The first question regarding the Wnt2 and Wnt9b double knockout mice. There's a very dramatic phenotype in the liver. I'm just curious whether this is largely due to developmental effect, or you think this could happen in the adult liver?
So it can happen absolutely in the adult liver. And although we have not inducibly knocked out Wnt2 and Wnt9b with tamoxifen or anything like that, I think what we have done is we have knocked out beta-catenin, for example, LRP56 with adeno-associated virus in adults. So if you use AAV8-TBG CRE in an adult animal, and you knock it out, within 3 days, you see glutamine synthetase going down. In 4 days, CYP2E1 goes down, so within 5 or 7 days. So what that really implies is that Wnt beta-catenin signaling is always on, and during your homeostasis, even if you're young or adult, and when you disrupt it, you're going to get this phenotype.
I see. That's great clarification. So my second question is about the windless IRAD CRE knockout model. So you did a lot of single nucleus RNA-seq analysis. You got a lot of clusters. I'm just curious, are those primarily hepatocytes, or you also got differential clusters for other hepatic cells?
Yes. So we did, for this particular knockout mouse model, we did single nuke-seq, and single nuke-seq enriches for hepatocytes, cholangiocytes, and some immune cells, not all. If we were to focus on immune cell compartment, it's better to do single cell sequencing, not single nuke-sequencing. So in this, we got, I would say, appreciable number of endothelial cells, and we got appreciable number of hepatocytes. So those were the two major compartments. We got little bit of immune cells here and there, but I don't think that we feel very comfortable with the amount of immune cells that we have to make big calls. So, but we are very comfortable with, because almost 80% of the cells that we got in this analysis were hepatocytes, and we were able to really, able to very carefully characterize the enrichment of different clusters between these two groups, and then able to validate some of them by immunohistochemistry or by molecular cartography for the 100 genes.
Yeah, that's very good. I just want to follow up on that, because you saw the communication between hepatocellular cells and endothelial cells.
Yes.
But you didn't identify a strong communication to hepatocytes. How could that regulate hepatic zonation? I don't know. Is this indirect to hepatocyte?
Yes, very, very good question. So I would say, Charlie, that I am fairly confident that a significant amount of this communication is indirect, meaning stellate cells are talking to endothelial cells and then directing them to express certain genes, which then are talking to hepatocytes. But based on cell chat, which is really an algorithm, right? It's a probability. It's not, it's not I wouldn't call it that it's a validation. This is hypothesis generating. Based on that, we did see stellate cells communicating to other stellate cells. We did see stellate cell communicating to hepatocyte, but the intensity of the signal was less as compared to the intensity between stellate cell and endothelial cell.
Great. Thank you so much.
Of course. Thank you for great questions, both of you. Yes.
Thank you so much, Charlie. Leah.
Thank you, Xiaoqing, and hi, Paul.
Hi, Leah.
Hi. So I have several questions. First of all, so a very interesting phenotype you see in your stellate cell knockout. This phenotype, I'm just wondering, because you see some body weight change is that you give a Western dye, do you see any sex differences, like female and male animal are the same response or different response?
So from the outset, I'll say that we only had 4 animals. So, these knockouts are very difficult to breed because, only the female knockouts, you can only use female LRAT Cree to breed for germline transmission. Males don't breed well. So, and the problem with that is anytime we are breeding females, we have to then utilize them. We have to kill them, right? Males, you can breed with females and then you can have more and more litters. Unfortunately, that's not the case. So with the limited number of animals that we had, we saw same phenotype. We saw none of the females had any steatosis, and all controls had significant steatosis in their livers.
I see.
After 3 months of Western diet.
I see. Okay. So second question is kind of I'm always want to figure out like a hepatocyte identity for the in terms of metabolic zonation in relation to liver regeneration. So basically, you have labor of division. When you have a proliferation, you cannot do function properly, right? So.
Yes.
Can you comment how this regeneration in terms of hepatocyte also have complication with Plody? So like you have like a diploid, tetraploid,
Yes.
and so is beta-catenin regulate both metabolic process, also regulate proliferation? Is it related to Plody or not?
That's a beautiful, beautiful question. So I can tell you, ploidy, we have not directly investigated. Andy Duncan, who's part of the Liver Center, we have talked about this many times, but we've never gotten a chance to look at the ploidy in our beta-catenin knockouts. I mean, the hepatocytes are smaller, but that doesn't mean anything, right? Yes, So I think I have been asked this question so many times that I better do it in the often, have lengthy discussion. The 3 or 4 groups who are dilemma. And what we believe is that there is some additional factor that is playing an important role. For example, if you have Wints alone, we think that they are not proliferation, they are differentiation. So if you have very high Wnt environment, which is there in zone 3, these cells never proliferate, really. So the paper that was published by Roel Noose in Nature in 2015 is completely correct. There's absolutely, because they used Exin reporter mouse, and Exin, when they made an Exin reporter, they knocked in one of the Exin alleles, and so they made a heterozygote for heterozygous for Exin. Exin is responsible for beta-catenin activity. So when you have an Exin hypomorph, beta-catenin is active, and in that case, then Jan and others have actually made better reporters with keeping the Exin function intact, and they don't see that, right? That zone 3 is proliferating. We never. If you have very high levels of Wnt. So, but now there's a gradient. But they also have something else. And those two we don't know what those we also know that beta-catenin alone doesn't cause cancer. So if beta-catenin has a second hit like MET, like RAS, or HERT, or NF-NRF2, then then those together, it independent. Remember that usually it's not wild type beta-catenin, right? Because in a normal zone 3, Wnt beta-catenin activity is balanced. You have Wnt, you also have axin. So it's almost like feedback mechanism, but only when beta-catenin is mutated and it cannot be degraded and you have a second hit. So then you get cancer.
That means when you have a hyperactive beta-catenin and the activity is always on and you get tumorigenesis.
That is correct, especially when there's a second hit associated with it.
Okay. Thank you very much.
Of course, of course.
Thank you, Leah. Julie.
Thank you, Xiaoqing. Hey, Dr. Mongo, fascinating job, and I really learned a lot. Yeah, so I have 2 questions. First, I know you just use around 100 genes probe to perform this study, fascinating study, and also the ligand and receptor crosstalk communication study analysis, kind of based on the cell studies. Not all the cell can be put out. One question is the immune cells zonation distribution. We know from the portal vein went to the central vein, so more immune cell, especially the macrophages or dendritic cell, is more outside the portal vein. So have you used histological analysis to see whether those immune cell distribution also changes in your models?
Yeah, good question. Very good question. So I can just tell you that not at the depth that we should have. So we definitely see immune cells are very different when beta-catenin or Wnt pathway is knocked out. And I didn't get a chance to present this data at all, but those of like Wenxing was there at ASIP a meeting. I presented on cancer, and Leah was there. And Leah's talk was right after mine, so she did get to hear, and I got to hear her beautiful talk. So what I was able to show there is when you have gain of function of beta-catenin, immune cell microenvironment becomes very different. So we actually get a lot of M2 macrophages. In addition, beta-catenin active tumors exclude CD4 and CD8 T cells. And that is why they don't respond to traditional immune checkpoint inhibitors. Like the first line of therapy nowadays is, these PD-L1 inhibitors, PD-1 inhibitors, CTLA-4 inhibitors. So they don't work very well when it's a beta-catenin mutated tumor. So I don't know if you guys know Yuli Wong, who's a wonderful scientist at Genentech. She was the first author on the 2022 Nature Medicine paper. And they showed in about, I don't know how many thousands of patients that they had on IMbrave 150, where they showed that beta-catenin mutated cancers were the ones that were not responding to immune checkpoint inhibitors. So again, a long-winded way of giving you the answer that although we have not directly looked at it, I think if you were to look at those immune cells in our knockouts, you'll find fascinating things. We just haven't had a chance to focus on that.
Thank you. My second question is for your HSC when knockout animals. I noticed the liver size kind of smaller, and the body weight seems also reduced. So because the question back to the donation distribution, bile acid is very important to the nutrient absorption. I'm just wondering from those 3, because it's secreted from row 3 and then go back to the row 1. And whether that one has the circulation from the zonation goes to the intestine bile acid secretion and then went back to the nutrient input, and whether those affect the liver functions.
Yes. So we looked at total bile acids, and we also looked at bile acid species. Xiao Chao Ma has a mass spec here in School of Pharmacy, and we gave them liver samples and blood samples. There was no real difference in the species of bile acid. Just the bile acid pool was increased. So I think what happens is when your zone 3 becomes broader, you are producing more bile acids. But then, as you can imagine, there is a lot of feedback inhibition, which may be playing a role in kind of trying to keep things as homeostatic as possible. But eventually, just because of the expansion of the zone 3, you do have a higher level of bile acid. So even if more bile acid goes into your intestine, you're absorbing more fat and more lipid. Now, our Wnt knockout hepatocytes seem to be capable of handling, lipids much better for fatty acid oxidation, for energy production. And I think that's why these animals are lean over time. But your point is well taken. Is it cause or effect? I think those are some difficult questions to answer. And I think as we get more and more number of mice, I think our goal is to kind of look at enterohepatic circulation of these bile acids, look at other parameters of lipid metabolism as well.
Thank you. Thank you very much.
Sure.
Thank you, Julie. The last panelist is Yang.
Hello. Thank you, Xiaoqin. Paul, that's a very great talk, and I learned a lot about the zonation. I'm really intrigued by this when you change the zonation that affect the liver regeneration ability and the systemic metabolism. So I wonder, can you really manipulate the zonation in the liver as a therapeutic approach to say improve the systemic metabolic profiling?
Yeah, I think that's eventually the end game for sure. Yeah. I think so. We are working with a company, Antelera, there are already another company, Surrozen, which is in San Francisco. And then Antelera is also in South San Francisco. And both of them have a Wnt agonist that they have used. We have personally used, it was actually in Cell Reports Medicine paper. It's a tetravalent antibody that binds directly to frizzles and LRPs to activate beta-catenin. So we gave that drug after acetaminophen overdose, and we also gave that drug after partial hepatectomy. And in both situations, we were able to improve liver regeneration. So, but we never looked at metabolic functions of that. Now, the question is, if you were to put high-fat diet animals and then give them this, would you be able to induce zone 3, induce fatty acid oxidation, and improve the regeneration and repair at the same time? I think that's a $1 million question, and I think that is very important to do. Now, remember that the drug that has been approved for use for MASLD is a thyroid receptor beta agonist. Right? So OCA doesn't work, or beta-cholic acid was not approved by FDA, but very recently thyroid receptor beta agonist has been approved. And back in 2012, we actually showed that thyroid receptor beta agonist activates Wnt signaling pathway. And it improves proliferation, it improves metabolism. So we do believe that if you were to give Wnt agonist, we would be able to induce both these activities, but we haven't directly tested that. And I think that's what the next steps should be.
Or instead of manipulating the zone itself, where we can learn the difference of the zone, then from there we can find some targets. That's possible.
That is also, that's another way to approach this. If you are normalizing, trying to normalize the zones, or if you are able to identify what zone specific changes, and then try to, manipulate that. I think all of these are fantastic, but almost every gene in the liver is going to be zonated. There are very few genes, even albumin, which is the largest, highest, one of the highest genes, is zonated. It's more in pericentral, more periportal than in pericentral region. So,
also, I like your concept. You said the zone that make liver kind of constrain the injury. So that make it repair better compared to other less regenerative organ. They kind of more homogeneous. So once that damage happens, they cannot constrain. Yeah.
Yes, exactly right. Yeah, I mean, I think that at least for drug-induced liver injury, DILI, this compartmentalization of injury allows the remnant cells to always persist for repair.
Yeah, thank you so much.
Of course, thank you. All very insightful questions. Really appreciate it.
Thank you, Yang. Dr. Morgan, we still have some questions in chat box. So, Dr. Bingo,
Hey, Paul.
you can mute yourself.
Hey, Ben's here.
Okay. Well, it's getting late. My question will be short.
Okay.
First, for your windiness, stellar cell knockout mice, I think, the phenotype, what do you think is the AR80 So it's not just, expressed in the hepatic stellar cells. There's other organs, lung, intestine, they express very high level.
Yeah. Yeah. I knew that somebody's going to ask me and call me out on that. Yeah. So I absolutely agree with you that the phenotype overall could be a net effect of all these organs crossing,
Winden, it's not just stellar cells.
That's right, yes. But, but we focused only on liver zonation. We only focused on liver mitochondrial function.
No, but I'm saying, this, high fat body weight, all this, that maybe is due to other organ.
I couldn't agree more. So guilty as charged.
Okay. Well, most people use the AR80, even like this AR80 DTR mice deletion, the mice looks very sick. I can tell.
Those are different. Yeah, I don't want to even use those for any physiological studies. The inflammation that happens when these cells die, the whole phenotype is very complicated. At least we don't see that.
Okay. Second question. I saw you one slide, APAP model. All this Ki67 cell hepatocytes surround the necrotic tissue.
Yes.
When we do Carney model, the hepatocyte proliferation seems pretty spread in the non-damaged area. Why does hepatocytes surrounding necrotic tissue in APAP model proliferate?
That's a very good question, and I can there are two ways to answer that question. One is through the lens of spatial cell single transcriptomics that was done by a group from Israel. They published in, I think it was Nature or Science or something like that just a couple of years ago. Maybe it was Cell Stem Cell, where they had looked at this particular model, and they showed after acetaminophen that and they don't go into the mechanism, whether it's damps that are being released, but the endothelial cells and macrophages that were present right at the interface of that injury were the ones that were upregulating the Wnt. So, how that happens, what is upstream of that, remains a very, very important question. Now, Udayan Apte, who used to be in my lab a while back and is now full professor here at KUMC with Wenxing and others, actually also showed that macrophages that are present in this surrounding area are somehow very sensitive to sensing that and upregulate. And Cynthia Zhu also showed the role of macrophages in Wnt upregulation. So, but what is the upstream effector? How do these cells sense? Remains a big question, but it's the area adjacent to the injury. So I'm very interested in finding, what did you guys find in your model, which leads to upregulation of whatever growth factors it is that you guys are looking at?
Yeah, we're trying to study, in our the paper last year, how this Carney model necrotic tissue resolved. We now study the APAP model. I think it's very different.
Yes. Yeah,
So,
I think
okay.
there's something with that, the area, the contact area where the necrosis happens. There's some magic going on there.
Okay, so I have last, quick question. The zonation you're talking about. These, all these liver have 3 zonations. All these zonation formation have have any benefits. For example, all these zone 3, why all these metabolic genes expressed in zone 3?
Yeah.
Not zone 1. I mean, does that express in the zone 3 have any advantage or benefit for the host?
Yeah, I think that's a good evolutionary relevant question. I think my understanding of this is it's not that everything is happening in zone 3. I think everything is actually happening in a very balanced way in all zones. For example, if you were to just even look at, glucose metabolism, then, gluconeogenesis is happening in your periportal in zone 1, whereas glycolysis is happening in zone 3. And, if you were to look at ammonia metabolism, protein metabolism, again, ureagenesis versus glutamine production. So I think it's like the liver is truly its division of labor. So there has to be, if one cell dies, one zone dies, because we all know ischemia can happen, xenobiotic toxicity can happen. And when one zone dies, at least the other zones will live and will either expand and retain the function, or we have done a study that is not published yet. We did partial hepatectomy and looked at global zonation, over 7 to 14 days. And what we found was zonation is maintained throughout the liver regeneration process. Never, ever did a regenerating liver lose zonation. Only all the zonated genes are now 3 times to 7 times, 10 times higher. But the zonation continues to be maintained. So to me, it tells that liver has this very intelligent sort of cell circuits that are maintaining its function. And I think there's no easy answer to this because it's such a big scale question that you asked.
Yeah, no, because zone 3 hepatocyte probably prefer to metabolize alcohol acetaldehyde.
Yes.
Why zone 3 have expressed as a high enzyme? Whether that help you metabolize faster or make you drink more. I don't know. Okay, this is just.
That's a good question.
Okay.
Yes, we will.
Okay, I stop here. Yeah.
Over a glass of wine someday.
Yeah. Well, good. Thank you, Paul.
Sure. Thank you.
Great talk.
Thank you. Appreciate it.
Thank you, Dr. Gao. Next question is from Yan Kai. Any ideas about upstreams of Wnt ligands? What regulates Wnt2 and Wnt9b in endothelial cells? What may regulate Wnt4 in stellar cells?
So I can tell you that Wnt2 and Wnt9b, that work is being actively pursued in our lab right now. So, we have a couple of knockouts that are brewing. We do believe that hypoxia is playing an important role. So we have HIF1, HIF2, and ARNT flox mice. So we are breeding these knockouts. And my hypothesis is that if you remove some of these hypoxia-inducible genes, you may affect the expression of Wnt2 and Wnt9b. Because remember that zone 3 is the most hypoxic zone in a liver lobule. It's 30 millimeter less. So in periportal area, the oxygen tension is 60 millimeter mercury. Whereas if you were to go into pericentral region, it's about 30 millimeter mercury. So that's now for Wnt4 in stellate cell. This is a very new area to us. So, I think that's a very important question. What regulates Wnt4? I don't have the answer to that at this time, Yankee.
Yeah, great answer. Thank you, Dr. Monga. But actually, yeah, we have done some studies. We found actually, it seems like HIF did not directly induce the wind. It's through some cells. But do you have an idea like something else, like a transcription factor or something, can directly bind to the promoter of wind ligand?
Yes.
Then induce it. And how, like, what's the difference between like Win2 and Win9B?
Yes.
Because they are all, both, yeah.
Yeah, beautiful questions. So this is my aim 3 of my R01. Okay, so I'm proposing all of these studies. So I think I'm on the right track. I looked at the promoter regions of both Win2 and Win9B. They actually have ARNT binding sites. They also have hypoxia response elements on them. But remember that, you know, it's not hepatocytes that are making Wnt. It's endothelial cells, right?
Yes.
So you have to knock out your hypoxia inducible factors from endothelial cells. And then the expectation is, so those animals are cooking right now in our lab. So hopefully in the next 2 to 3 months, you know, we should have an answer, you know, at least some preliminary evidence of that. But then we will see once the grant comes through and everything else, if they're going to fund me to do all of this or not.
Sure, look forward to your work. Thank you. A very great talk.
I appreciate it. Thank you.
Thank you, Yankai. Dr. Paul Huang.
Yes.
Yeah, you can unmute yourself.
Hi, Paul. This is Paul from Shanghai Fudan University. Very great talk and learn a lot from you and so much information. It really pushed the liver field forward. I'm not doing liver research, but you know, from perspective of lymphatic biology, people believe that lymphatics are almost everywhere, right? But in the liver, people probably don't think that it function very important. Because as you said that the blood vessels are very sinusoidal, right? So it permeabilize everything, so it doesn't really require the functional lymphatics. Although people, for example, Colorado University, people showed that in pathophysiological condition, lymphatics is there. And then my question is that you in many couple of places you use a LIVE-1 Cree to delete the endothelial cell. In you know, to my knowledge, that's lymphatic endothelial, which, as I said, that it's very different from blood endothelial cell. But then your conclusion jumps to endothelial cell. So I was also lymphatic LIVE-1 Cree also expresses in the macrophages and also fibroblasts. So to me, is that possible? Your results suggest that there's a niche effect of Win secreted by macrophages, also fibroblasts, to to regulate the zonation in addition to blood endothelial cell. Maybe you should use the Cree created by Nancy Speck, CDH5, which is more specific. Can you comment about it?
Yes, that's a great question, and relatively guilty as charged. So I can tell you that LIVE-1, we absolutely agree that it's a lymphatic vessel endothelial cell. That's what the name stands for. But if you were to take LIVE-1 and you look at any of the endothelial cell markers, sinusoidal endothelial cell markers in the liver, you take CD31, you take c-KIT, you take FEBP4, any of those, there is actually a spot on co-localization. So the sinusoidal endothelial cells of liver are very strange endothelial cells. They are not traditional vascular endothelial cells. As I mentioned, there's no basement membrane, there's no other features. And on top of it, you have large fenestrae. Under electron microscopy, you can actually see these beautiful openings. So, the good news is that when we use LIVE-1 Cree, we did reporter mice to kind of confirm that. So we are not saying that we are not deleting it from lymphatic vasculature within the liver, which is gaining more and more importance, right? Yasuko Iwakiri from Yale is doing that work. There's another person, Sanjukta from Texas A&M, who's looking at lymphatic vessels in the liver. So, but when we do lineage tracing, it was very clear to us that all vascular endothelial cells. Very interestingly, we looked at macrophages. We looked at co-localization with macrophages. We looked at co-localization with the stellate cells. We did not find any of the lineage tracing in those cells. So maybe the Cree lines may be functioning slightly different than what actual life would be expressed in. The other gratifying thing to us was the two other groups published almost simultaneously with us, and they used VE cadherin Cree, and another group used stabilin-2 Cree, and which was more endothelial specific, and the results were completely the same as us. So while I agree with your point in principle that some of the phenotype may be somewhat different, because it may be a more niche type of a deletion. Based on all the analysis we have done so far, it seems highly unlikely. But unless we do it, as you said, with Nancy Speck's mouse or some of these other, in fact, my reviewers have always told me to use an inducible Cree. But for the sake of convenience, I'm just using it, what I have in-house, because all my reporter analysis has shown very clear endothelial cell penetrance.
Yeah, thank you. So you also use Lysam Cree, right? Maybe I missed it. Can you remind me about the data of the Lysam Cree?
Yes. So Lysam Cree, we looked at zonation.
Right.
Again, using traditional markers like CYP2E1, CYP1A2, glutamine sensitive, no difference. That was a hepatology paper in 2014. And then we looked at liver regeneration, and we found a 24-hour delay in liver regeneration, like the stellate cell Wnt knockouts, but it was not as profound as when you knock out beta-catenin from liver regeneration, from the liver. It was also a milder phenotype as compared to when you knock out Wnt2 and Wnt9b from endothelial cell. So we think most of the Wnts for regeneration and for zonation are still coming from endothelial cells.
Got it. Yeah. Okay. Thanks so much. Yeah. Very nice.
Nice to meet you. Yes.
Okay. Our next question is from Ping. Is how the zonation is dysregulated in MASH and associated with the impairment of the hepatocyte regeneration?
Yes, that's again a good question. And I think a simple answer, without having directly investigated this myself and based on the literature, is that the zonation is really maintained in MASH and MASLD. It's only that the lipid deposits that happen are zonated. So you first see in zone 3 and zone 2 and then in zone 1. So there is expansion of that. But if you were to look at zonation genes, for example, glutamine synthetase or CYP2E1, they will still show very zonated patterns. So I think that I wouldn't go as far as saying that zonation is messed up. I would say that the lipid accumulation and as a result, lipid peroxidation and reoxidative stress is zonated. But zonation, I wouldn't call it to be dysregulated. And definitely the regeneration is impaired, because it has been shown by many groups that as these liver cells accumulate fat, they become very poor at differentiating, at proliferating. It's almost like senescent associated phenotype, as well as increased cell death and injury. So I will kind of leave it there, without going too much into the detail.
Interesting. So next question is from Luo Chao at Peking University. So Wnt from endothelial cell regulates hepatocyte beta-catenin signaling and liver donation. How does the Wnt influence endothelial cell itself? Is changing in endothelial cell the primary cause of liver donation regulation or secondary?
Yeah, a good question. It's a complete chicken or egg story. So I think the easiest answer for me is we don't know. But my belief is that hypoxia is regulating the expression of Wnt within endothelial cells, but then we have to prove it, right? But then how does Wnt influence endothelial cell itself? I think that remains a good question, which we'll have to knock out some of the other compartments, Wnt, to kind of look at their crosstalk with endothelial cell. But remember, I have already shown you data today that if you are knocking out Wnt from stellate cells, the endothelial cell Wnt expression changes. So how Wnt4 is inhibiting Wnt2 expression in endothelial cells, it remains a big question, in my mind. And is it possible that Wnt4 is causing degradation of Wnt2 somehow through some mechanism that we don't understand yet? I think those are the kind of questions that we are asking.
Yeah, great. So maybe last question. Let's see. I know it's kind of late.
That's okay. That's all right. That's all right.
So this is kind of more than comments. Like a great talk, Paul. Is from Haitao Guo.
Yeah, I know Haitao very well. He's from Pittsburgh, right? So
yes, Haitao, are you here? Yeah. So let me read the.
Yes, I'm here, but
go ahead.
It's too late.
Haitao, how are you?
Yeah.
Sorry, I missed the talk of your student today because I wanted to come over there to the Cancer Center at noon, but then I had a 1 o'clock meeting. I had to be physically present, so it was not
okay.
Possible.
Understandable. So yeah, she did a good job today. Actually, she can catch up with you later. Yeah,
that sounds great.
Right.
Please send her my.
Yeah, I have talked to you before about this. So actually, zone 3, the central zone area actually is a comfortable zone for HBV.
Yes,
HBV actually replicates better there. So there, when the beta-catenin pathway, some of the transcription factors is helping the virus to transcribe the RNA. So that's a perfect match.
Yeah. So remind me, Haitao, it's the receptor for HBV is BSEP, right?
No, it's NTCP. It's
I'm sorry, NTCP. Yeah, NTCP. So NTCP is zonated.
That one I don't know. I think that's only about the transcription level. It's not on the entry level.
Okay. Okay. So I mean, obviously this is a hypoxic zone. So is it possible that HBV, depends on glycolysis products or hijack some of the other machinery?
There's some report on that from Jim McCain at Oxford University. Yeah, that's a couple of papers, but nobody else has repeated that. So it's still
we have Wnt inhibitor that works on human hepatocytes. So if you want to do some cultures, we also have beta-catenin knockout mice. I know you have some versions of your virus which will work on mouse hepatocytes, but we also have human hepatocytes and an inhibitor of beta-catenin. If you want to use those and inhibit beta-catenin and see if HBV are unhappy, then I think that would be important.
I know that you always be generous. Yeah. So yeah, you want to keep me busy, so I can do that.
You will get your 5th R01 then.
Yeah, that's true. Yeah. Good talk. Yeah.
Thank you for joining. I appreciate it, Haitao.
Okay.
Yeah. I don't see additional questions. Xiaoqing, do you have anything to say?
Well, I actually have a very naive question. Because Dr. Morgan, when you present the wild type and lockout mice, I just wondering how old they are. Because you do mention at the very beginning, the aging can contribute to the liver mortality and morbidity. And when you start the zonation, I just wondering, the age of mice you use.
Yes, that's also a very, very good question. And, just for the sake of simplicity, we have always analyzed our animals between 2 months to about 4 months. because, it's too young. If you have two young animals, like, for example, 1 month old, the zonation is not fully established, so I don't think that anybody has very carefully looked at every single, so if I had a lot of money, then I would have done, molecular cartography. It costs a lot, like $20,000 to do 8 samples, right? So if I had different time points, like 5 days, 10 days, 15 days, 20 days after birth, to look at these 100 genes and then just see how a zonation is established postnatally. But when we have looked at just a few markers here and there, some markers for zonation are there at very early, like 10 days. Like glutamine synthetase, for example, is very much apparent. But if you look at some other markers like CYP2F2, or you look at HAMP, they are not there until almost 2 months after birth. So I think zonation is a very dynamic process, and the whole, sort of patterning is happening up until 2 months of age. So we have not really done studies beyond 6 months of age. So I think your point is very important and relevant, that will we get the same phenotypes if we were to examine these animals at 1 year of age? I think that's a good point. The only problem is you have to keep these animals alive for, no graduate student wants to do that. No postdoc is going to do that. So it remains a very difficult. So just for convenience sake, I think people are doing this in relatively short time frame.
Yeah. Paul, I have maybe a question.
That's true. It's really, yeah.
Thank you.
Yeah, go ahead, please.
Yeah. So when I talk with Haozhou about his tracing thing, he said zone 2 migrated to zone 3. Actually, when you check the marker, actually the cell from zone 2 becomes zone 3 marker.
Yes.
So
Yes.
it's kind of like environment in trend.
Yes.
So my technical question is, I think you have beautiful work to tracing the spatial RNA expression. So environment before the tumor is like epigenetic play a really role to remodeling this transcription machine. So in addition, like molecular cartography to tracing gene expression, is there any way to tracing like an enhancer or something epigenetic?
Yeah, people have done single cell ATAT-seq.
Yeah.
And you can, barcode your samples now. If you label with any of Haozhou's Cree, for example, right? You can label periportal cells or pericentral cells with GFP or TD tomato, etc., and then do ATAT-seq, with injury, without injury. But again, very expensive experiment, as you can imagine. Single cell sequencing and ATAT-seq is not going to be cheap. It's going to be $20,000-$25,000. So I think everybody is kind of believing that it's environment that is playing a role. So you take cell out from one environment and place it in another environment, then that hepatocyte will start expressing the genes that are relevant to that environment. And it's because the macrophages, the stellate cells, the endothelial cells that are there in that environment are actually dictating what the hepatocytes should express. And again, we and others have done this work very nicely. Acetaminophen, like, for example, simple, you have this hole in the liver in zone 3, but as these cells migrate, they start to express zone 3 marker. Glutamine synthetase becomes positive. CYP2E1 becomes positive. So it's all environment. But what is the epigenetic state? That's the hardest question that you can ask. Unfortunately, yes.
Okay. Yeah.
Yeah.
Thanks
The only last other thing would be laser capture microdissection. So people are now beginning to use, again, a lot more laser capture microdissection to ask these types of questions. So if you have a good laser capture microscopist amongst you may be able to kind of do some, it will be almost bulk RNA type work, but you can cut your zones based on, the location.
I see. Last one question for the last one. this kind of benefit for this 88 people audience still in the room. I together with Sylvia, I think your code did also provide service if people want to do the molecular cardiography, right? So
Yes.
people, if you want to do this measure, talk to Paul.
Yes, I'll be very happy. Resolve Biosciences is actually improving its platform, and they are going up to 500 genes. We were able to only do 100 genes, but they are able to do 500 genes and add 50 antibodies. So, and they told me, I was just on a call with them yesterday, and they told me that they are able to do it also on paraffin sections. So all my studies have been done on fresh frozen livers, but they said that now with their advanced technology, which is available starting July 1, they'll be able to do FFPE. So if you just do Resolve Biosciences, I don't have any shares in the company. I don't have any money coming to me. I just love this technology because I have been able to see things that I was not able to see before. So if anybody else can benefit from it, I think it'll be great for scientific community.
Thank you so much.
Well, thank you so much for, again, keeping me here and for such lively discussion. It was truly very nice to see a lot of people. So stay in touch. If I can be of any help, anytime, email me, talk to me. I'll be always available.
Yes. Yeah. Okay. Yeah.
Thank you very much, Paul.
Thank you very much.
Thank you.
Okay,
Thank you
very
so
nice
much,
talk.
Thank you.
Dr. Monga.
Oh, when you still here?
Yeah.
Wow.
There's one other question here in the Yangui. Hello, could you please help me ask one question? Could Paul just give a brief summary of the relationship between liver zonation, cholesterol, and endothelial cell? That's complicated. Liver zone, cholesterol and endothelial cell. So that's I don't completely understand the question.
Doi, can you see the question?
I did not see the
can't
question
see
in
the
the
question.
public chat.
Oh, I think he by mistake he sent to me.
Okay.
But yeah. So so it's from Xiao Cheng. I'm probably not pronouncing it right, but I will just put this to you guys, and then maybe I can answer it if you guys give him or her my email. Okay. Then this is that question that came to me
I see.
from Yangui.
Okay. Just give a brief.
A brief summary of the relationship between liver zonation, cholesterol, and endothelial cells.
Yeah. I mean, so that's
why cholesterol is here.
Yeah. I think so cholesterol definitely, cholesterol is what converts, is used to form bile acids. So cholesterol is metabolized in zone 3 to give rise to bile acids, but they are sequentially, sort of formed from pericentral towards zone 1, and ultimately they're secreted in the bile. So if you look at all bile acid metabolism genes, they are located mostly in zone 3, very little in zone 2, and very, very low in zone 1. So I would probably just sort of leave it there. And there's a lot of literature on it, on bile acid metabolism and zonation. So if you want to look that up, just do a search. And if you have more directed questions, I'll be very happy to answer them. Okay.
Yeah, fascinating. Yeah, thanks. Yeah, I see pretty late. Have a good weekend.
Okay. That's all right. Thanks, guys. Very nice to see you.
Yeah.
Take care.
Thank you.
Bye-bye.
Bye.
Thank you. Thank you so much for your time. Yeah. Have a good one. Yeah.
You too.
Bye.
Bye.